Testing. Testing. Okay, that works. All right.
Recording in progress.
And the snow just keeps coming. BJ and his crew are out removing snow, and now it looks like they hardly did anything. It just keeps coming and coming and coming. All right, gang, it is time to begin. Hope you guys are doing well. I know you guys don't like Zoom, but the administration has given us instructions. It's not my job to tell the administration to go pound sand. It's my job essentially to teach within the parameters that they provide. So they said we're going to Zoom today, so we're going to Zoom. Here we are. We will be back on the saddle tomorrow. If you don't have a lab book, new lab books were ordered. They were done. They are on the Mrs. Schaefer's desk in the natural science office. If you paid for one, just go in and get one. If you haven't paid for one, please get one and then leave a note saying that you picked one up, or tell someone so we can write it down so that we know that you owe us at least for the lab manual. So lab, for those who have lab tomorrow, we will meet in 103. Very good. Let's have a word of prayer and then let's begin. Lord, your word tells us that since we call upon a Father who judges each person's work impartially, we should live as foreigners here in a way essentially that pleases you. Father, help us now to live lives that please you. Thank you for this snowy day. And we pray even through Zoom that you would help us understand how cells work so we can understand how tissues are put together since they're made of cells, and then eventually how organ systems work. In Jesus' name, amen. All right, we ended up last time talking about some of the membrane-bound organelles. We looked at the lysosome. Lysosomes are membrane-bound. They contain these acid hydrolases. The membranes of lysosomes consist of these so-called V-type, V as in victory, V-type ion pumps that pump protons into the interior of the lysosome, thus acidifying them. That acidification of the lysosome activates the acid hydrolases, and now those acid hydrolases can degrade proteins, complex carbohydrates, lipids, glycolipids, things like that. The acid hydrolases are only active at a low pH. And here is a lysosome in a transmission electron microscope picture. You can see it here. So if we pull out our little colored pen here, this is the lysosome here. And notice it's in close proximity to a peroxisome. And MB stands for not microadivich, but microbody. Microbody means it's a small vesicle inside the cell and you have no idea what it does. That's usually what microbody is. Microbody also is used to mean something that comes from the endoplasmic reticulum. Here's another lysosome. You'll notice how electron dense these structures look, and that's really common for lysosomes. They look really electron dense. Now their partner in crime over here, the peroxisome, that structure is going to contain enzymes that help oxidize unusual lipid-soluble molecules. It also contains catalase, which will degrade hydrogen peroxide to water and molecular oxygen. So peroxisomes are involved with degrading strange lipids. They're also involved with detoxification of reactive oxygen species. Liver cells, for example, will have lots of peroxisomes since they're involved with metabolizing many drugs and other xenobiotics our body comes into contact with. Xenobiotic just means foreign chemical, that's all it means. And that leads us to the energy factory of the cell, the mitochondria. Mitochondria have two lipid layers, two membranes. The outer membrane is relatively permeable when you compare it to other membranes. The inner membrane is extremely tight and extremely impermeable. The inner membrane also has a lot of protein embedded into it, far more than other types of membranes. Inside the inner mitochondrial membrane is a compartment called the matrix, just like in the movies, I guess. And the mitochondria make this molecule called adenosine triphosphate, or ATP. ATP is the energy money of the cell. If you want to make something, you have to pay for it. What's the dollars you use? You use ATP. So that's your energy currency. And ATP, the lion's share of the ATP in the cell is made in mitochondria. So here is a mitochondrial structure. Again, this is shamelessly stolen from your book. You have an outer membrane here. It's relatively permeable. There's the outer membrane. You then have the intra-membrane space in between these two. And then you have, let me get a different color here. And then you have your inner mitochondrial membrane here. And then this here is your inner mitochondrial space. Within the inner mitochondrial matrix of membrane, of course, is the matrix. Soluble within the matrix are ions, metabolites, ATP, ADP, also various enzymes. And then another thing you have within the matrix is you have your mitochondrial genome. You actually have DNA molecules. Those DNA molecules are circular. There's many of them usually available at one time. And the organization of these genomes is very bacterial. They're organized into operons. They get expressed like operons, and it's a very bacterial looking molecule. You also have within the matrix ribosomes, mitochondrial ribosomes. And those ribosomes, for all intents and purposes, are very bacterial. I'll just say ribs there, ribosomes. So the proteins that are expressed, the messenger RNAs that are made from this mitochondrial genome is translated on ribosomes in the mitochondria that also look very bacterial. So some of the proteins, definitely not the majority, but some of the proteins involved in the activities of the mitochondria are not encoded on the nuclear genome, but are encoded on the mitochondrial genome. Mutations in the genes found on the mitochondrial genome cause these very interesting and very deleterious mitochondrial genome diseases. And those mitochondrial genome diseases tend to be very severe because they're the result of mutations in the mitochondrial genome. They show a so-called maternal form of inheritance. That is, it doesn't matter what the genome of your father is, it matters what the genome of your mother is. You inherit this straight through your mother. And this is hard to write with the mouse. Maternal inheritance. I had one of these iPads and I had one of those pens, I could just write it with my lousy handwriting instead. Maternal inheritance. Another membrane structure that is contiguous with the outer nuclear membrane is the endoplasmic reticulum. The structure of the endoplasmic reticulum can vary from cell to cell. For some, it looks like a highly folded, kind of crepe paper looking bunch of membranes. In other cells, the endoplasmic reticulum can look almost tubular. There are two types of endoplasmic reticulum. There is a studded looking endoplasmic reticulum known as rough endoplasmic reticulum. Those studs that decorate the ER are ribosomes that are in the process of protein synthesis. So these will serve as the site where you synthesize proteins that are going to be housed in vesicles, secreted or trafficked to various membranes within the cell. So the cell membrane or certain internal membranes too. You also have smooth ER. Smooth ER has enzymes for cholesterol and lipid synthesis, and also groups of enzymes for detoxification of molecules. A very interesting group of enzymes called cytochrome P450s. So here's rough ER. Again, you can see it's contiguous with the outer leaf of the outer layer of the nuclear membrane. It's studded with ribosomes that are in the process of protein synthesis. And then smooth ER, which has a bit more tubular look to it. And this houses enzymes for phospholipid synthesis, cholesterol synthesis, and cytochrome P450s that detoxify molecules. And that leads us to the ribosomes. These can be attached to the ER or free within the cytoplasm. These are the sites where protein synthesis occurs. Ribosomes are complexes of ribosomal RNA and ribosomal proteins. They have two subunits, a small ribosomal subunit and a larger ribosomal subunit. You can see that here. The darker color is the large ribosomal subunit. The lighter colored entity is the small ribosomal subunit. The large ribosomal subunit in human cells is 40S. The larger ribosomal subunit is a so-called 60S. Structure. Together, they form a so-called 80S ribosome. Now, the first thing you're probably thinking is, well, wait a minute. 40 plus 60 is 100, not 80. You're absolutely right. S stands for Svedberg. Svedberg. A Svedberg is a unit of descent within a centrifugal field. Macromolecules descend in a ultracentrifugal field based on their size and their shape. When the ribosomal subunits come together, they now have a shape that doesn't descend as fast as the two of them separately. So they make a structure that's more compact, that's more streamlined, and you don't get the drag in the centrifugal field on the basis of their shape. So they will get a little more hung up in the field. They'll therefore have an 80S size, not a 100S size. 80S is significant because bacterial chromosomes are 70S. And the ribosomes from mitochondria are also 70S as well. Here is what's called a Miller spread. This comes from Art Miller, who was at the University of Chicago for years. He recently retired. Well, recently, 5-6 years ago. And you have a RNA molecule here, a messenger RNA molecule. You have ribosomes essentially on that messenger RNA molecule. And these ribosomes are in the process of making proteins. So you can see these are lined up very nicely. The proteins here are bigger than the proteins over here. That tells you that translation is occurring in this direction. And this shows you essentially ribosomes at work making their newly synthesized proteins. Incidentally, I forgot to mention this. If you have questions, do please ask questions. Try to use the chat function on Zoom to ask questions. And if I don't see it, or if you don't like using the chat function, just unmute yourself and go ahead and butt in, so to speak, and ask questions. I'm fine with that. All I ask is that if you're not asking a question, please mute yourself so that what comes through your speakers doesn't then come out into my speakers and cause feedback, because that will be bad for everybody. All right. So just a quick pause here, make sure everybody's good. No questions. No? Those ribosomes that are on the ER are making proteins that will be loaded into vesicles, or proteins that will be inserted into the ER membrane, in which case they'll still be loaded into vesicles, but the protein will be floating in the membrane of the vesicle. And then they'll be delivered to the Golgi. And if they're native resident ER proteins, they'll be trafficked back to the ER from the Golgi. From the Golgi, then they'll either go to the membrane or be loaded into secretory vesicles or go to other types of structures or be released as lysosomes or whatever. Here's a very beautiful picture, again, shamelessly stolen from your book, of a ribosome in the process of translation attaching to the endoplasmic reticulum, making the protein. The protein undergoes co-translational modification by the attachment of trees of carbohydrate to aspartic acid residues, asparagine residues. These trees are pre-made, then they are attached to asparagine residues by an enzyme called the oligosaccharide transferase, the OST. These proteins, these trees will be trimmed later on as the protein goes to the Golgi, but these trees are going to help proteins in the ER fold the protein. That's what these trees are for. They are for helping the protein fold. They also tell the ER how long the protein's been there and whether folding it is a lost cause, it should be degraded, or whether new carbohydrates should be attached so it can be refolded. How does the ribosome get to the ER? It starts off in the cytosol. If it's making a protein that's supposed to go to the ER, there's a sequence at the amino terminus called it, well, your book calls it a leader sequence. It's actually called a signal sequence. Signal sequence. That signal sequence then gets bound by a RNA protein structure called a signal recognition peptide, or signal recognition particle, as it's sometimes called, the SRP. The SRP escorts this and it attaches to the ER. It plops down the ribosome. When the SRP binds the ribosome, the translation stalls. It plops it down. There's a pore on the ribosome that is a complex made of a protein called Sec61. And then once the SRP leaves, translation resumes, and the protein then is extruded into the ER lumen. And if the protein is supposed to go on the membrane, it will actually leave the pore and then end up being in the membrane, something like that. Once it gets made in the ER, that protein then is trafficked in a vesicle to the Golgi apparatus. The Golgi apparatus and the ER are kind of a 1-2 punch for delivering packages of proteins and things like that. The Golgi then will sort the proteins, send them to their appropriate location and what have you. So think of the ER and the Golgi as kind of the FedEx, if you will, of the cell. Here's a really beautiful TEM picture of the Golgi. You can see it's a bunch of flattened disc-like membranes. And you have vesicles essentially that are coming to and leaving the Golgi. So this part of the Golgi here from which things leave, this is called the trans-Golgi network. That sounds like a television station, doesn't it? Trans-Golgi network. These flattened vesicles, these are called cisternae. Cisternae, C-I-S-T-E-R-N-A-E, cisternae. And these are vesicles essentially that are coming here. And you can see these vesicles, they're starting to fuse over here. They're fusing, and then these are going to eventually all fuse and make their own cisternae here. And then these cisternae will mature as they go in this direction, eventually becoming trans-Golgi network. The ER and the Golgi are also sites of lipid synthesis. Lipid synthesis begins in the ER. Those lipids are loaded into a vesicle. They're sent to the Golgi. The Golgi finishes the synthesis and then sends them to their appropriate sites. Cholesterol is made in the ER. It's made in the ER and then it's loaded into lipoprotein particles also in a compartment of the ER. The proteins of that compartment, however, get made in the ER and get sent to the Golgi. To make the lipoprotein particle, they get sent back to the ER. So here is a little cartoon, again, shamelessly stolen from your book. You see the nucleus, you see the ER, Golgi apparatus. These two work in concert with each other. Some of the vesicles that come from the Golgi become lysosomes. Others are secretory vessels that will fuse with the plasma membrane. And there you are. All right, that is the end of that. Are there any questions before we move on to stages of mitosis and all that? If there are no questions, then we'll go ahead and take a look now at the second part here. Let's talk a bit about how cells divide. So the so-called cell theory has two planks in it, namely that all living things are composed of cells. And then cells come from pre-existing cells. That's the second plank. You don't have cells being created by some sort of chemical precipitation process. Cells become fathers, if you will, by making daughter cells through the process of mitosis. The cell theory is often credited to the two German scientists, Schleiden and Schwann. They came up with the first plank in that. They're often credited, at least in high school biology books, for coming up with the second plank as well. That's actually incorrect. The person responsible for the second plank in the cell theory, namely that cells come from pre-existing cells, is Rudolf Virchow, the father of pathology. Rudolf Virchow came up with that second plank by studying tumors. And in fact, Virchow was one of the pioneers of tumor biology. Now, this process called mitosis often refers to cell division, but technically speaking, mitosis refers to nuclear division. Nuclear division. The actual division of the cell is cytokinesis. So can you have mitosis in cells without cytokinesis? The answer is yes. That's how you get multinucleate cells. So proper cell division includes mitosis with cytokinesis. It was thought for a long time that mitosis and cytokinesis were inextricably linked. Several different laboratories have used various types of cell-free and cell systems. Probably the biggest mover and shaker of this has been John Newport at UCSD to show that those two processes are separable. Meiosis occurs in germ cells. Meiosis is used to make gametes. So whereas mitosis makes two cells that are genetically identical, meiosis takes a cell that is diploid. It takes the diploid number of chromosomes, duplicates it, and then that cell divides twice to make 4 cells that are haploid. So meiosis is a reductive form of cell division that produces a haploid gamete from a diploid progenitor. So if human cells have 23 pairs of chromosomes, 46 chromatids, you duplicate that so that you have 46 chromosomes, 92 chromatids. You then divide once. So you have 2 cells now with 23 chromosomes. Those 2 cells then divide again, and the chromosomes separate into chromatids. And now you have 4 cells, each with 23 chromatids. They're haploid. And again, meiosis only occurs in cells that will make gametes, germ cells. Human cells, just like all other eukaryotic cells, go through a so-called cell cycle. A cell cycle is highly coordinated, highly complex. It prepares the cell to replicate the DNA and then divide. The cell cycle has been extremely heavily studied, largely because abnormalities of the cell cycle are a hallmark of cancer. And that's why the cell cycle has been so heavily studied. Our knowledge of the cell cycle and the things that drive it have led to the production of particular types of therapies now that interfere with elements of the cell cycle that are used to treat very fast-growing, very aggressive types of tumors, like melanomas and pancreatic cancers and things like that. And while these treatments don't cure those cancers, they definitely slow them down much better than old treatments used to. And maybe in the future, we'll have a cure for these types of cancers. So the cell cycle consists of these four processes, and these four processes are very highly coordinated. The first is the growth of the cell. In order for the cell to divide, it has to be big enough in order to give equal amounts to the two daughter cells, so those daughter cells can flourish. So the cell has to grow. And the cell won't properly divide unless it's big enough and has enough volume. The second is DNA replication. The cell has to replicate its DNA, make sure the DNA replication occurred properly, fix it if it didn't go properly, and then be able to distribute those duplicated chromosomes into daughter cells. And then to properly divide those cells, that cell, so that you get to daughter cells with the right number and complement of chromosomes. Those are the four processes you have to coordinate in the cell cycle. So you might remember from high school biology, mitosis consists of interphase, prophase, metaphase, anaphase, telophase. And my really dumb mnemonic for remembering this is, I parked my army truck. I parked my army truck. Interphase, prophase, metaphase, anaphase, telophase. I parked my army truck. Yes, it's dumb, but hey, if it helps, then use it. Now, interphase is broken into three phases. An interphase is the longest phase in the cell cycle. If the cell cycle was 24 hours long, just to put a number on it, 23 of those hours would be spent in interphase and 1 hour would be spent in mitosis and cytokinesis. So this should underscore the importance of interphase and how long it is. So after mitosis, interphase begins with the first G phase, G1. Now, some papers or books will say G stands for gap. Others will say G stands for growth because the cell is growing during this time. Gap, growth, it's G phase. I don't care if it represents gap or growth. It represents one of those two or both of those two. So there, how's that? So during G1, the cell is in fact growing. That's an important part of G1. And you can see the centrioles are going to replicate during G1. They don't move a whole lot, but they replicate. In fact, they replicate, and it's actually hard to tell that they've replicated because they're so close together. From G1, you go to S phase. This is a really important phase. So with S phase, you're going to take your DNA and you're going to synthesize it. So you're replicating DNA. This is the only time during the cell cycle when you will replicate DNA. Once the DNA is properly replicated, you move into the second G phase, G sub 2. This is a phase that prepares the cell for mitosis. And that begins with the onset of prophase. Now, if the cell wants to go from G1 to S to G2, it needs growth factors. Growth factors essentially are going to drive this cycling through the cell cycle. If you get to G1, you get to this boundary here and you don't have enough growth factors, the cell is going to drift off into a form of cellular sleep called G sub 0. G sub 0. And that G sub 0 is a quiescent phase. You have many cells in your body right now that are in G sub 0. For example, if you damage your skin, fibroblasts that are asleep in G0 will be awakened by molecules made by inflammatory cells. They will wake up through those growth factors, one of them being platelet-derived growth factor. They will then go to the area of damage. They'll start cell cycle anew. They'll pass through. They'll become into S phase. They'll divide. They'll get to the site of damage and they will make collagen to heal the damage. And this G sub 0 state is called a post-mitotic state. A post-mitotic state. Memory cells of your immune system, your memory B and T cells from that COVID-19 vaccination or flu vaccination you might have received. Those cells are in G sub 0. They will enter G1 very rapidly, though, if they're activated by the proper antigen. And here's a drawing that I did to show you G1, S, G2. G0, again, this is this quiescent state. If you get the right trigger, you can re-enter G1 and then pass go and collect $200 and so on and so forth. Now, for those of you who've seen this in the microscope, this will be familiar. For those who haven't, you'll see it tomorrow when you look at onion root tip. Chromosomes of cells that are in interphase. They are not easy to see. You also may have a nucleolus. Centrioles are too small. You won't see those with a light microscope. Once you pass into prophase, at the beginning of prophase, the chromosomes now will become highly condensed. They become short and thread-like. You can see them. Centrioles will start to move apart. You start to get microtubules called spindles that will emanate from them. They will move to either side of the nucleus, and then the nuclear membrane will break down. These chromosomes now will be available to these spindles, and some of these spindles will bind to the kinetochores that assemble on the centromeres of the chromosome. Let me go to our blackboard here. So let's just pretend this is a chromosome here. So this structure here is the centromere. Not all chromosomes have their centromere toward the center. Some will have it up here like this. Others will have it at the end there. It varies from chromosome to chromosome. I've drawn it here just for convenience. So the centromere is a special part of the chromosome. This is wound into very tight chromatin. That very tight chromatin is called heterochromatin. And on this heterochromatin, you're going to assemble a protein complex. That protein complex can bind the DNA of the centromere, and it can also bind the microtubule. This protein complex is called a kinetochore. So the kinetochore kind of acts like an adapter, One end of it will bind the DNA of the chromosome. The other end of it will bind the microtubule. Which is that's what makes the spindle. And this, of course, is centromeric DNA. And that's essentially what's going to capture those microtubules from the spindle. So these microtubules here that capture the kinetochore, those get called kinetochore microtubules. Kinetochore microtubules. You'll have one microtubule from either side here, bound to either side of the chromosome pair, chromatid pair chromosome here. They'll provide tension on those chromosomes. When that tension is equal, the chromosome is going to line up at the equatorial plate here. When all the chromosomes are properly captured by these kinetochore microtubules, and then providing proper tension on each chromosome so that they all line up at the equatorial plate, that's the onset of metaphase. The nuclear membrane is completely disappeared. Spindle fibers have captured the chromosomes properly. There are other spindles that are extending out this way. Those are called asters. A-S-T-E-R-S comes from the Latin word for star. Then there are a few other of these microtubules. And these are going to essentially go this way, and they're going to overlap. And those are going to be a different type of microtubule, spindle microtubules. And those spindle microtubules are going to provide force that are going to drive these two poles in opposite directions. After metaphase has come on board, eventually, the bonds that hold the chromosomes together are dissolved. The chromosomes are held together by proteins called cohesins. Cohesins act like little molecular handcuffs. When those molecular handcuffs are degraded, that tension on the chromosomes pulls them apart. And when the chromosomes are pulled apart, that is the onset of anaphase. You pull the chromosomes apart, and then these two poles actually themselves move further away. And then eventually, and you can see in their wake, there's this material here that's left. Those are your spindle microtubules. They're going to make a structure called a midbody. That midbody is going to be important here in just a second. The midbody now is going to provide a scaffolding for the assembly of a band of actin and myosin. That band of actin and myosin is going to pull these two parts of the membrane together in what's called a cleavage furrow or a division furrow. The nuclear membrane, when it dissolved into little vesicles. Those vesicles actually bind to the surface of the chromosomes. If you take the chromosomes out during this time, the nuclear membrane will not reform. They need the chromosomes to do this. They bind to the surface of the chromosome and they fuse into little nuclear vesicles around each chromosome. Those little vesicles fuse And then eventually you get a makeshift nuclear membrane surrounding these chromosomes. And so your new nuclear membrane forms, your nucleolus then will gradually reappear. You get the formation of a cleavage furrow, and that is the onset of telophase. And there are these sites that show these very beautiful pictures of mitosis. I invite you to look at those. And then cytokinesis involves essentially these two parts of the membrane touching, fusing, and the separation of these two cells. That is cytokinesis. Meiosis is very much the same as mitosis, with the exception that you have prophase, metaphase, anaphase, telophase 1, and then prophase, metaphase, anaphase 2. Prophase 2 commencing after telophase 1. The other difference is in the way these chromosomes line up at metaphase. Here, these line up in this fashion so that essentially these chromosomes will separate. It's different for meiosis. For meiosis, as you can see, with metaphase, the chromosomes, instead of being lined up one on top of the other, they line up side by side. And so the bonds that hold these together are broken. These will then, well, the bonds that hold them together actually are the places where the chromosome is crossed over, the chiasmata. Those get resolved by topoisomerases, protein called REC8. These then separate during anaphase. So the chromosomes separate during anaphase 1. During anaphase 2, the chromatids separate into their separate cells. And that's how you get 4 daughter cells that are haploid. So the big difference is the way the chromosomes line up at metaphase 1. In meiotic cells, the 2 chromosomes are linked together by crossing over, because recombination has occurred during the very complicated prophase 1. You get the formation of something called the synaptonemal complex between the chromosomes. That synaptonemal complex holds them together. The synaptonemal complex is the scaffolding for the assembly of something called recombination nodules on them. The recombination nodules catalyze crossing over. The crossovers themselves hold these home chromosomes in place before the onset of anaphase 1. That's the big difference between meiosis and mitosis. So it's really prophase 1, which is the money phase, that's different between mitosis and meiosis. Homologous chromosomes line up side by side rather than one on top of the other. And so this shows you meiotic prophase 1, very complicated process that has several substages. There's a leptotene, pachytene, diplotene, diokinesis, so on and so forth. And yeah, chromosomes pairing, they cross over, and then, yeah, that's the way that goes. All right. Any questions about cell division? You'll see mitosis tomorrow. You won't see meiosis. We don't have any, well, scratch. We have some testes and ovary. You might see cells undergoing meiosis in the testes. That's where you'd see them, if you look carefully enough. My wife just tested me. Western schools have decided to close tomorrow. So I'm going to bet that the plows will be out because the snow is supposed to stop by tonight. We're going to have clear roads tomorrow. That's what I'm betting. But I'm not a betting man, so just don't listen to what I said, okay? They're my predictions there. My predictions, yeah, that and, you know, $10 will buy you something at Starbucks. Very good. With the time remaining, let's go ahead. Oh, stop. Let me stop there. Does anyone have any questions about cell structure? I realize that was review for many of you. Doesn't hurt to review. There might be some new things there. Is there anything that was unclear? No? All right, let's talk about how cells use and make energy. So lots and lots and lots of chemical reactions occur inside cells. Lots of them. Tons of them. Untold thousands. Those reactions are largely catalyzed by proteins called enzymes. There are some enzymes that are RNA molecules. For example, in ribosomes, the large ribosomal RNA is the ribozyme that catalyzes the formation of a peptide bond on the surface of a ribosome. There are also ribonucleases that have an RNA component. And the RNA component is actually the business end of the ribonuclease. For the most part, though, entities that speed up the rate of reactions in cells are enzymes, are proteins. So enzymes are protein catalysts. Remember, catalysts, they speed up chemical reactions without themselves being ultimately changed. So they're not consumed, they're not changed during the reaction. They increase the reaction rate. The reaction rate. They don't change anything inherently intrinsic to the reaction. So they don't change the equilibrium constant for the reaction. They change the rate to the reaction, and that's it. How do enzymes do this? They do this by stabilizing the transition state of the reaction. Virtually all reactions have an energetic hurdle they have to leap over. That hurdle, that energy required to get over this hurdle is called the activation energy. Enzymes essentially stabilize the transition state and lower the hurdle. That's why the reaction can go much faster, because the energetic hurdle it has to jump is much lower. That's how they accelerate the rate of reactions. So here again, this is taken from your book. So this constitutes the energy of the reactants here. Those are your reactants or energy up here. Here's your products, the energy is up here, but you've got this very tall wall that you're going to have trouble climbing. The enzyme is going to take this wall and give it a haircut. So now this can just roll right over this, and down it goes. And that can essentially speed up the rate of the reaction. So here the activation energy is this much, over here it's this much, just to illustrate this. The reactants of the reaction will bind to the enzyme at a specific site. That site is called the active site. The reactants that bind to the enzyme are called the substrates. The fact that the substrates bind to the active site is responsible for the specificity of the reaction. And once the reaction has been catalyzed, you generate a product. The product does not have a high affinity for the active site. So the product gets released from the enzyme. So you can write an enzyme catalyzed reaction in this fashion. The enzyme plus the substrate gives you an enzyme substrate complex. That enzyme substrate complex produces a product, and the product dissociates from the enzyme. Now you have a free enzyme and a product, and the enzyme is free to go back here, redo this again, pass go, collect $200, and win the Monopoly game. And this, once again, is stolen shamelessly from your book, but you can see. So you have substrate A, substrate B. Notice how they fit the active site, they bind. You get a product. That product now doesn't have a terribly high affinity for the active site. They dissociate. Now the enzyme, free enzyme, is free to do this all over again. Bottom line, the enzyme remains unchanged at the end of the reaction. Might be changed during the reaction, but it gets changed back to its original state. So it's ultimately unchanged. So we might write ultimately in there to make this a bit more accurate. Some enzymes bind one substrate. Other enzymes bind multiple substrates. The binding between the enzyme and the substrate, as we said, is quite specific. The enzyme will take the substrates, it'll orient them so that they can react and form the transition state. Then it takes that transition state and stabilizes the heck out of it so that it will degrade to the product rather than just going, ooh, I don't want to form that, I'll just be the reactants again. So it gets over that energetic hump and becomes the product. Once it becomes a product, it dissociates from the enzyme. How does the enzyme do this? It binds the reactants using side chains of its amino acids, because remember, the enzyme's a protein. So it's a polymer of amino acids. Those amino acid side chains form transient interactions, low energy bonds with the substrate. It orients them so that they can react from the transition state. Those amino acid side chains will further stabilize the transition state, so that then it can become the product. Now we'll do a experiment that will demonstrate this, but enzymes have environmental optima. They have temperature optima, they have pH optima, they have salt concentration optima. Some enzymes do much better with higher concentrations of protein in the environment. Others don't care. Some enzymes prefer to interact with membranes in order to catalyze their reaction. Others don't care. So enzymes have certain environmental optima in which they work. So this, for example, is ribonuclease. Ribonuclease, as you can see, has its peak optima for temperature right here. That optima is somewhere between about 37 degrees and somewhere around about 42 degrees or so. So it's a pretty narrow temperature optima. After that, its ability to catalyze the reaction really decreases. Also, if you get colder, its tendency to catalyze reactions also tends to go down. These enzymes show their different pH optima. Pepsin, which is an enzyme that works in the stomach, which is very acidic, has an acidic pH optimum. Salivary amylase, however, works the best at strongly neutral pHs because it works in the mouth, where the pH is relatively neutral. Trypsin, which is made by the pancreas and is secreted into the duodenum, now works with large amounts of bicarbonate, which are made by the pancreas as well. So it has a basic pH optimum, and that's demonstrated here in this as well. So enzymes have certain environmental optima in which they work. Outside those optima, they don't work as well. Many enzymes are just amino acid polymers. Sorry, they don't need anything else in order to catalyze their reaction. Other enzymes, however, have, in addition to the amino acid polymer, small molecules that are either organic molecules or they may have metal ions. And these extra molecules are called prosthetic groups. Prosthetic groups. And they assist in catalyzing the reaction. So here's your enzyme, for example, here are your substrates, and here's this prosthetic group. If this prosthetic group is an organic molecule, then it's usually called a cofactor or a coenzyme. This enzyme here is called an apoenzyme. Apoenzyme. The enzyme here with its prosthetic group, that's called a holoenzyme. Holoenzyme, apoenzyme. Okay. All right, my computer is ready to die. The battery's almost out, so we're going to stop right there. We'll pick this up on Friday. Don't forget Friday, we have a quiz. And I will hang for just a bit. Oh, how much battery power do I have left? I have 12 minutes of battery power. So I'll hang after we finish. If you have any questions, please hang out and I'll answer your questions beyond that. I hope you have a great evening and I'll see some of you at least tomorrow at 8 o'clock for lab. Okay? Ciao, gang.
Dr. Baranovich,
Yes.
for the quiz, is it going to be what we covered recently or from all of the above?
All the above.
Okay. All right,
So
thank you.
chapter 1, and we skip chapter 2, and then 3 and 4 are just 3. I think it's 3 and 4. I need to check that. But the enzymes and energy one, I think, is chapter 5. Let me check that. So I think it's 1, 3, and 4. So those are the ones you should review for the quiz, okay?
Okay, thank you.
No problem.
And then is it the quizzes or the exams that are based off the questions in the back of the
The
chapters?
quizzes.
Okay, sounds good. Thank you.
All right.
Question.
Yeah, no.
What's the difference between a cofactor and a coenzyme? Is one organic and the other inorganic?
Coenzyme usually is meant to refer to an organic molecule. The thing is, some organic molecules will have metals in them. So the designation is not crystal clear.
Okay.
So prosthetic group is a nice term that kind of covers the whole thing. That's why I kind of like that term. So coenzyme can refer to metals,